Hi everyone, my name is Rachel Babb, and today I will be showing you the poster I've made over the past semester. I was looking at designing an mRNA vaccine candidate for SARS-CoV-2, specifically looking at variant P.1 with a mutation located at amino acid 614 of the spike protein. So the SARS-CoV-2 virus was first identified back in 2019, and since it was discovered, a variety of different variants have emerged. One of those variants is the P.1 variant, the one studied here, and it emerged in Brazil originally. Our current data suggests that these mutations have an effect on antibody-mediated neutralization, making it more pathogenic. Currently, we don't know if the Pfizer, Moderna, or J&J vaccines provide any immunity for this variant, and so I designed a new mRNA sequence for variant P1 with mutation at 614 of the spike protein in case the vaccines do prove ineffective. Um, this sequence could then be used as a template to increase efficiency in our response to an outbreak if it does prove ineffective. So a couple things to note. The first is this here is a linear protein map containing the wild type SARS-CoV-2 spike protein and a bunch of different genes that are important. I will point you over here in a little bit to show you where the mutations I made are located. Additionally, here are the amino acid substitutions that I made to transform the wild type sequence into the P1 variant that is applicable for a vaccine. And lastly, over here, we have the wild type spike protein, a PyMol model that I designed. So the main aim of this study was to design a potential vaccine candidate for the P1 vaccine. And how we did this, I started with the wild type mRNA sequence and performed 2 rounds of mutagenesis to lock the protein in its closed conformation. So basically the spike protein has 2 different conformations, one that's open and one that's closed. The closed one is more antigenic, and so it's the one that we are targeting with this vaccine design. In order to lock the spike protein in that conformation, um, 2 mutations had to be made at amino acids 986 and 987. You can see those right here. This was transforming lysine and valine into proline mutations. This was done because proline is an inflexible amino acid, and that allows the conformation to stay locked close. The second round of mutagenesis, you can see the primers here again, was making that mutation at amino acid 614. This was an aspartic acid mutation into glycine to make this specific for the P1 variant. Once this sequence was designed, it was inserted into a vaccine design containing a T7 promoter, 5' and 3' UTR regions, and a poly A tail. These are all necessary for stability and effectiveness inside the body. This new design was then inserted into a PGEM3Z vector plasmid using restriction enzyme EcoR1. This was used for in vitro transcription, and you can find the procedure that was performed right here. After about 4 hours of transcription, the plasmid and sequence were purified by HPLC. So following purification in the future, if this sequence was to be used for a vaccine, a nanolipid particle would need to be designed to contain the RNA and delivered into the body. Additionally, testing would need to be done to determine the effectiveness of this vaccine against the P1 strain and the safety for the human body. Thank you so much for taking time to listen to my poster today. If you have any questions, I'd be more than happy to talk with you. Feel free to reach out, and I hope you have a wonderful rest of your day.